So today we are discussing about a topic in the embryology, that is fertilization. So how fertilization takes place and what are the effects of fertilization. So we know that the fertilization takes place in the ample of the uterine tube. The fertilization takes place in the uterine tube. We know that on either side of the uterus we have uterine tubes are present. We know that on either side of the uterus we have uterine tubes are present. This is the uterus, and on either side of the uterus we have uterine tubes are present. So in the ample of the uterine tube the fertilization takes place. So the secondary oocyte as well as the the secondary oocyte as well as spermatozoa, they have to reach the fertilization site, that is the ample of the uterine tube. Though the secondary oocyte from the ovary and also the spermatozoa, they have to reach the ample of the uterine tube. So how the approximation of the gametes takes place. So when the sperms are deposited in the vagina, what happens here is the sperms release contains a enzyme called prostaglandin. The sperms contain an enzyme called prostaglandin. This prostaglandin increases the contractions of the uterine muscles. This prostaglandin increases the contraction of the uterine muscles, and also the oxytocin, which is secreted by the neurohypophysis, that is nothing but from the hypothalamus. The oxytocin from the hypothalamus, which are stored in the neurohypophysis, it also causes the contraction of the uterine musculature. Then what happens is that due to the contraction of the uterine musculature, what happens here is due to the successive contractions of the uterine muscle, they sucks the sperm. They sucks the sperm which is deposited in the vagina during the coitus. Okay, and because of the prostaglandin, which increases the uterine contraction of the muscles, as well as the oxytocin, which also increases due to this successive contraction, what happens here is they aspire the sperms. They aspire the sperms due to the successive contractions of the uterine muscles. And here, at single ejaculation, 300 to 500 millions are deposited. At single ejaculation consists of 300 to 500 millions of sperms. Out of these 300 to 500 millions of sperms, only 300 to 500 sperms reach the ovum. Out of these, so out of the 300 to 500 millions of sperms deposited at one ejaculation, only 300 to 500 sperms only they reach the secondary oocyte. And what about all these? What about these remaining millions of sperms? We know that the cervix consists of abrupt constrictions. We know that the cervix of the uterus, we know that the uterus consists of fundus, body of the uterus, and also cervix, right? The cervix of the uterus consists of abrupt constrictions which doesn't allow the spermatozoa to enter into the uterine cavities, because the out of these 300 to 500 millions of sperms, only thus the remaining sperms, they are blocked due to the abrupt constrictions of the cervix. And also at the uterine ostium, means where the uterine tubes opens into the uterine cavity is the uterine ostium, and the uterine ostium also the sperms are obstructed. They doesn't allow the sperms to enter into the uterine tube. And one is the abrupt constrictions of the cervix as well as the uterine ostium. It doesn't allow the sperms to enter into the uterine tube. That's why only 300 to 500 sperms only they enter into the uterine tube to the place of fertilization, and the remaining sperms, they are obstructed. They are obstructed by the abrupt constrictions of the cervix as well as the uterine ostium. And coming to the, out of the 300 to 500 of sperms, only one sperm fertilizes the oocyte. Only one sperm fertilizes the mature ovum. So out of these 300 to 500 billions of sperms, these 300 to 500 billions of sperms, they are arranged over the secondary oocyte. They are arranged over the secondary oocyte, and these sperms, they have to enter into three obstructions. They have to enter into the mature ovum by passing through the three areas. One is corona radiata, then the next one is zona pellucida, and the next one is periviternal membrane. These cells are called corona radiata cells. corona radiata cells. So the sperms have to pass through corona radiata and then they have to pass through zona pellucida. This is zona pellucida. And they have to pass through perivitelline membrane. Perivitelline membrane. Okay, so the sperm has to pass through all these barriers, all the protection barriers of the oocyte. They have to pass all through the 3 protection barriers of the oocyte. One is corona radiata. So the sperms, one sperm enter into how the corona radiata cells they disintegrate. They disintegrate by an enzyme called hyaluronidase. Hyaluronidase secreted by the acrosomal cap of the sperm. We know that the sperm head, it is covered by an acrosomal cap. The sperm head, it is covered by an acrosomal cap, right? Which is secreted by the Golgi apparatus. The sperm head, it is covered by an acrosomal cap, which is secreted by the Golgi, which is formed by the Golgi apparatus. And this acrosomal cap secretes one enzyme called hyaluronidase. Hyaluronidase enzyme, it disintegrates the corona radiata. It disintegrates the corona radiata, and also all the remaining germs also helps in the disintegration of the corona radiata. And only the compatible sperm only it enters, it reaches the zona pellucida, the next protective barrier of the secondary oocyte. The first protective barrier of the secondary oocyte is the corona radiata, and the hyaluronidase enzyme secreted by the acrosomal cap, it disintegrates the corona radiata, and the sperm now enters, it reaches the zona pellucida. And now what happens here is the sperm now releases an proteolytic enzyme called acrosin. Now the sperm releases a proteolytic enzyme called acrosin that disintegrates the glycoprotein layer, nothing but the zona pellucida. So, the proteolytic layer that is called acrosin. This is the acrosin. And now, what is the third barrier? The third barrier is the perivitelline membrane. Now, the sperm has to pass through the perivitelline membrane. Now, what happens here is the head of the sperm fuses with the perivitelline membrane by two disintegrine peptides. By the two disintegrine peptides, the head of the sperm only enters into the perivitelline membrane and fuses with the perivitelline membrane. Thus, the sperm enters into the secondary oocyte. Now what happens here is the secondary oocyte completes its second meiotic division and converts into a mature ovum. So only after the entrance of the sperm, only the secondary oocyte completes its second meiotic division and converts into a mature ovum. Before entering, it doesn't complete the second meiotic division before its entry of the sperm. Only after entering of the sperm, only the secondary oocyte completes the second meiotic division and converts into a mature ovum. So the sperm head has to pass through 3 obstructions. One is corona radiata. These are disintegrated by hyaluronidase enzyme by the acrosomal cap, and the next one is zona pellucida, which is disintegrated by the acrosin enzyme, proteolytic enzyme, which disintegrates the zona pellucida, and the third one is perivitelline membrane, which is disintegrated by the 2 disintegrine peptides. Okay, now the sperm head enters into the perivitelline space. The sperm head enters into the secondary oocyte zone. Now what happens here is the oocyte undergoes the second meiotic division and the second polar body enters into the perivitelline space. Okay. Now what happens here is we know that the male pronucleus, and this is the female pronucleus, and this is the male pronucleus. The sperm head, that is enlarged and forms a nucleus that is called female pronucleus, and the mature ovum also enlarged and it forms a nucleus, and it arranged in the form of a nucleus called female pronucleus. They are also called ootids. And here what happens here is the DNA is replication of the DNA takes place, and see here fusion of the male pronucleus and female pronucleus, right? Male pronucleus and female pronucleus takes place, and the centrioles of the sperm enter along with the sperm. These centrioles, they are arranged at the opposite poles. Now what happens here is achromatic spindle takes place. Achromatic spindle takes place along the centrioles, achromatic spindles takes place and the chromosomes are linearly arranged along the achromatic spindle during the metaphase. Now what happens here is the cleavage division completes and two blastomeres are formed. Two blastomeres are formed. Actually, the actual self-fusion does not take place, only the linear duplication takes place. Now the male pronucleus and female pronucleus, they unite and they form a zygote consists of diploid number of chromosome, and they form a zygote consists of diploid number of, you know that 22 plus X from the haploid number of chromosomes from the mother, and 22 plus Y haploid number of chromosomes from the father. It may be 22 plus X also, because of the millions of sperms, half are X carrying and half are Y carrying chromosomes. Half sperms they carry X chromosome, half sperms they carry the Y chromosome. If the X chromosome carrying sperm fertilizes the ovum, the resulting is the female child. If the Y chromosome carrying sperm fertilizes the ovum, the resulting child is the male. So it further decides the sex of the embryo, further decides the sex of the child. Okay, so that's about the fertilization. So here what happens here is the DNA replicates and here the achromatic spindles are formed and centrioles are arranged in opposite poles, and the achromatic spindles are, the chromosomes are linearly arranged along the achromatic spindles during the metaphase. Now what happens here is the meiotic division, the mitotic division completes and two blastomeres are formed. Now we know that these blastomeres, they are multiply and form, and the 16th cell stage we call it as a morula, and we call it as blastocyst. We have learned them during the formation of bilaminar and trilaminar germ discs. Okay, so that's about the fertilization. So the fertilization takes place first year. The out of 300 to 500 millions of sperm deposited in the vagina, the uterine contractions of the muscle, it is stimulated by the prostaglandin as well as oxytocin. Due to the successive contractions, they suck the sperms into the uterine cavity, and here the abrupt constrictions of the cervix as well as the uterine ostium, they doesn't allow more sperms to enter into the uterine tube, and the sperms they are get blocked by the abrupt constrictions of the cervix as well as the uterine ostium. And now only out of 300 to 500 millions of sperms, only 300 to 500 sperms enter into the uterine cavity, and also from the ovum, the secondary oocyte also enters into the uterine tube by the ciliary beads of the uterine tube as well as the musculature of the uterine tube, and they reach the ampoule of the uterine tube. How the fertilization takes place here is because the sperms are linearly arranged, the sperms are arranged over the corona radiata, and the sperm head it has to pass through three barriers, the three protective layers of the secondary oocyte. One is corona radiata, it is integrated by a enzyme called hyaluronidase enzyme, and the next one is zona pellucida, which is also disintegrated by a enzyme called proteolytic enzyme called acrosin. These are secreted by the sperm heads, and also two disintegrated peptides which also disintegrates the perivitelline membrane. That the sperm head fuses with the perivitelline membrane, and now the sperm enters into the uterine cavity, and here what happens here is the secondary oocyte completes its second meiotic division and converts into mature womb. Here the DNA replicates, and what happens here is the centrioles along with the sperm head, they reach the opposite poles and the achromatic spindles are formed, and the nucleus, the DNA replicates into from one end DNA to two end DNA. Here diploidic number of chromosomes acquired, and here half from the mother and half chromosomes from the father, and the mitotic division completed and two blastomeres are formed. So that's about the fertilization. So today we are discussing about placenta. So we know that the blastocyst which is formed, we know that how the blastocyst is formed. The zygote undergoes repeated division and it forms a group of cells called morula, and this morula they are divided into inner cell mass and outer cell mass, and these inner cell mass we call as embryoblast and outer cell mass we call it as trophoblast. We know that. And how this trophoblast enters into the uterine cavity and enters into the uterus, and it gets implanted at the junction of the posterior wall of the fundus and the body of the uterus. Okay, now what happens, how the blastocyst gets implanted with the zona pellucida, it disappears and the blastocyst get implanted. Okay, now how placenta? So we know that this is the uterine endometrium. The uterine endometrium we call now as decidua. When after the blastocyst gets implanted in the uterine endometrium, the uterine endometrium is in the secretory phase. The stromal cells, they are large and vacuolated and they are filled with heavy drops of lipid and glycogen. And it is in the secretory phase. The uterine endometrium, we call it as decidua. The place where the placenta is formed, this place we call it as decidua basalis. And the part of the endometrium which separates this is the uterine cavity. This is the uterine cavity. The part of the endometrium which separates the uterine cavity from the blastocyst, this endometrium we call it as decidua capsularis, and the remaining part of the endometrium is the decidua parietalis. So the endometrium where the placenta has to be formed is the nothing but the decidua basalis. Okay. Now what happens here is, we know that the trophoblast is, this is the trophoblast, inside the trophoblast we have extra embryonic mesoderm is present. The trophoblast, we dealt them this trophoblast and the extra embryonic mesoderm during the formation of bilaminar and trilaminar germ discs. So we know that the trophoblast along with the extra embryonic mesoderm, we call it as chorion. This layer we call it as chorion. So various villi project into the decidua. Various villi they project into the decidua. These villi are called as chorionic villi. First, the chorionic villi, they project all over the trophoblast. This chorionic villi, they project all over the trophoblast. Now what happens here is, the chorionic villi over the decidua capsularis, they disappeared and we call it as chorionic levee. We call it as chorionic levee. Okay, chorionic levee. So, and the chorionic villi which projects towards the decidua basalis, we call them as chorionic frondosum. We call them as chorionic frondosum. The main units of placenta are nothing but the villi. What are villi? So we know that the villi, how the villi are formed. The villi are of three types. Primary villi. The main functional unit of placenta is villi, and these villi are of three types: primary villi, secondary villi and tertiary villi. We know that the trophoblast, it is along with the extra embryonic mesoderm, we call them as chorion, and the villi projecting from here we call it as chorionic villi. In the primary villi, we have the trophoblast undergo divisions and it forms syncytial trophoblast. Plus the trophoblast consists of a single layer of cells. The trophoblast consists of single layer of cells. This is called cytotrophoblast. Now what happens here is, these cytotrophoblastic cells, they undergo division and they form mass of cells which lose their cell walls, we call it as syncytiotrophoblast. Syncytiotrophoblast, and this is the cytotrophoblast. Okay, syncytiotrophoblast, and this is the cytotrophoblast. So the cytotrophoblastic cells, they undergo division and they form a mass of cells called syncytiotrophoblast. So what are primary villi means? The villi consists of syncytiotrophoblast as well as the cytotrophoblast. We call them as primary villi. And what are secondary villi? But secondary villi consists of, these are cytotrophoblastic cells, and we have mass of syncytiotrophoblastic cells are present which lose their cell walls. And here we know that beneath the trophoblast we have extra embryonic mesoderm is present. So extra embryonic mesoderm is present here. Okay, so these are cytotrophoblastic cells and these are syncytiotrophoblastic cells. So the villi consists of syncytiotrophoblasts, cytotrophoblasts as well as extra embryonic mesoderm. These three constitutes the secondary villi. So one more layer is added in the secondary villi. Syncytiotrophoblasts, cytotrophoblasts as well as the extra embryonic mesoderm constitutes the secondary villi. Primary villi constitute only syncytiotrophoblasts and cytotrophoblasts. Secondary villi constitute syncytiotrophoblasts, cytotrophoblasts as well as the extra embryonic mesoderm. Okay, and coming to the tertiary villi. Tertiary villi, they consist of cytotrophoblast, syncytiotrophoblasts, extra embryonic mesoderm, and they are traversed by the fetal blood capillaries. These constitute the tertiary villi. So what are tertiary villi? Tertiary villi consists of syncytiotrophoblasts and cytotrophoblasts, extra embryonic mesoderm and fetal blood capillaries. They constitute the tertiary villi. Fetal blood capillaries constitute the tertiary villi. And we know that the cytotrophoblast consists of mass of cells. Right, the cytotrophoblast consists of mass of cells. And what happens here is this is the cytotrophoblast, and the syncytiotrophoblasts consists of mass of cells, right? Then what happens here is the lacunae appears in between the cells of syncytiotrophoblasts. Later what happens here is these lacunae, they are regularly arranged. Now these are lacunae which are regularly arranged in the intervillar species, and we have syncytiotrophoblastic is present here. And within the syncytiotrophoblasts we have cytotrophoblast is present. Here, and this cytotrophoblast goes over the syncytiotrophoblasts and forms a plate called cytotrophoblastic cell. Okay, so these are the intervillar spaces, and these are in between. Those villi are consists of syncytiotrophoblasts, cytotrophoblasts, some extra embryonic mesoderm is present, and also fetal blood capillaries are present. And the intervillar spaces, they are filled by the maternal blood. Okay, because as the syncytiotrophoblasts goes towards the endometrium, what happens here is the maternal blood capillaries, they get ruptured, and the intervillar spaces, they are filled with the maternal blood. So when we take one section of the villi, so this is the intervillar space, right? And the villi consists of syncytiotrophoblasts, right? Syncytiotrophoblastic is present here, and then we have cytotrophoblastic is present. Okay, cytotrophoblastic is present surrounding the villi. Cytotrophoblastic is present, and later on we have extra embryonic mesoderm is present. Which I am represented with blue color. Okay, which I am represented is blue color. Here another intervillar lacunae is seen, intervillar space, and here we can see syncytiotrophoblasts, and we have after the syncytiotrophoblasts, cytotrophoblast is present, right? This is cytotrophoblast which grows over the syncytiotrophoblasts, and we have Beneath the cytotrophoblast, we have extraembryonic mesoderm is present. Okay. Extraembryonic mesoderm is present. And now we have the fetal blood capillaries are present here. Fetal blood capillaries are present here. So the villi consists of from inside to outwards. So the villi consists of fetal blood capillaries, extraembryonic mesoderm, cytotrophoblast and syncytiotrophoblast. And here the lacunae or intervillous spaces, they are filled by the maternal blood. Maternal blood. And now what happens here is the exchange of gases takes place through these villi, the fetal blood capillaries. The exchange of gases takes place here, and also the exchange of nutrients also takes place here, because the maternal blood it supplies the nutrients, water and electrolytes to the fetal blood, and here the fetus also it excretes urea waste products through the maternal blood through these villi. Okay, through these villi it excretes the urea waste products into the villi and into the intervillous spaces. And also here what happens here is the developed fetus, it consumes about 20 ml of oxygen per minute. In developed fetus consumes about 20 ml of oxygen per minute. This barrier is a protective barrier. The placenta, it acts as a protective barrier, and it doesn't allow any microorganisms to enter into the fetus. And also, but sometimes the poliomyelitis viruses and rubella viruses, they may enter into through the placental barrier, but most of the bacteria, they get obstructed by the placenta. And here the IgG and IgE immunoglobulins and IgG immunoglobulins, they pass through the placental barrier. Okay, they pass through the placental barrier. IgG immunoglobulins mostly, they pass through the placental barrier. And also here what happens here is some of the viruses may enter, and some of the drugs also they enter to the placental barrier. They may be fatal to the fetus. Okay, so the villi consists of first syncytiotropoblast, cytotrophoblast, extraembryonic mesoderm and fetal blood capillaries. Here exchange of food materials and exchange of gases, exchange of nutrients and exchange of gases takes place through these villi. Okay. So that's about the placenta. And here sometimes the placenta consists of many lobes. We call it as a lobed placenta. And one more thing is, when we look at the placenta from outside, it consists of cotyledons. These are 50 to 22 cotyledons are present. And how these are maternal cotyledons. How the maternal cotyledons are formed means, the endometrium sends trabeculae. The endometrium present here, it sends trabeculae into the intervillous spaces, and this constitutes the one maternal cotyledon. This constitutes the maternal cotyledon. So the endometrium sends trabeculae into the intervillous spaces, and these constitute the maternal cotyledon. They which are separated by grooves. When we are seeing from outside, they consist of grooves, and these trabeculae are separated. This trabeculae constitutes the grooves. There are 50 to 20 cotyledons are present. And here one more important thing is, these villi, they undergo branches. These villi they undergo branching. They divide into these called anchoring villi. These anchoring villi again divides into branches. They are called ramus corae, and this ramus corae again divides into branches called ramulus corae. So the villi, they undergo branches and branches. We know that how villi are formed. The villi are formed consists of syncytiotropoblast, cytotrophoblast, extraembryonic mesoderm and fetal blood capillaries. So these villi, they undergo branches and they form they are called truncus corae, ramuli corae and ramulus corae. They divide into branches and branches. Okay, and one such villus and its branches forms the fetal cotyledon. And the maternal cotyledon, how it is formed is, the endometrium sends septa into the intervillous spaces and forms the maternal cotyledon. So Coming to the abnormalities of the placenta, sometimes the placenta is lobed, we call now the placenta as the fetal placenta, and sometimes the placenta is diffused. Sometimes the placenta is diffused. That means the villi, they are present all over the villi, they are present all over the blastocyst. The villi, they are present over here all over the blastocyst. That time we placenta, we call it as fused placenta. And sometimes what happens here is the placenta, separate placenta is present here. Some separate placenta is present here, some placenta tissue is present here, and we call it as placenta succenturia. And these are the some normal abnormals of the placenta. And one more thing is, usually the placenta gets attached to the upper uterine segment. What is upper uterine segment? We know that the uterus divides into fundus, body and cervix, right? The uterus divides into fundus, the uterus divides into fundus, body and cervix. The fundus and upper part of the body of the uterus calls upper uterine segment. The fundus and upper part of the body, it forms the upper uterine segment, and the lower part of the body and the cervix forms the lower uterine segment. And this is the internal os. This is the internal os where it communicates with the vagina. Okay, the uterus communicates with the vagina through internal os. So fundus and upper part of the body forms the upper uterine segment. Fundus and upper part of the body forms the upper uterine segment, and lower part of the body and cervix forms the lower uterine segment. Usually the placenta gets attached to the upper uterine segment. Upper uterine segment. Okay, and sometimes the placenta gets attached to the lower uterine segment, and we call that condition as the placenta previa. We call that condition as the placenta previa. Placenta previa. If the placenta gets attached to the lower uterine segment other than upper uterine segment, we call it as placenta previa, and this placenta previa, it may cause problems during childbirth, and it may also fatal to the fetus. Okay. So here placenta previa consists of 4 degrees: first degree, second degree, third degree and fourth degree. Here the placenta previa the placenta gets attached to the lower uterine segment. The placenta gets attached to the lower uterine segment but does not reach the internal os. The placenta gets attached to the lower uterine segment but does not reach, this is the internal os, right? Where the cervix communicates with the vagina through the internal os, through an opening called internal os. So when the placenta gets attached to the lower uterine segment but does not reach the internal os, we call it as the first degree of placenta previa. Now, say in the second degree of placenta previa, the placenta reaches the internal os. The placenta reaches the internal os but does not cover it. The placenta reaches the internal os but does not cover it. That is called second degree of placenta previa. Then and the third degree of placenta previa is the placenta reaches the internal os and covers it. But what happens here is due to the parturition, what happens here is the internal os expands and the placenta does not over the internal os. That does not overlap the internal os during the parturition and does not cause problem somewhat. This is the third degree of placenta previa. And coming to the fourth degree, which is very, very dangerous, the placenta completely covers the internal os. The placenta completely covers the internal os. That is the fourth degree. That is the fourth degree of the placenta previa. In the first degree, the placenta does not cover the internal os, but in the second degree, the placenta reaches the internal os but does not cover it. And then the third degree, the placenta covers the internal os but it occludes during childbirth. That is, it covers a part of the internal os only, the third degree. And in coming to the fourth degree of the placenta previa, the placenta completely covers the internal os and it's very dangerous. Okay. And coming to the sites of implantation, you know that the implantation usually takes place at the first at the upper part of the body of the uterus, fundus and the body of the uterus and the posterior wall. But the implantation may takes place in the uterine tube. It is very dangerous to the mother. It causes profuse bleeding to the mother, because if the implantation takes place in the uterine tube, what happens here is, as the zygote grows and grows, the embryo grows, what happens, it causes the rupture of the uterine tube. So it causes the rupture of the uterine, it causes heavy bleeding, is fatal to the mother. And sometimes the implantation occurs in the lower uterine segment. Usually the implantation occurs in the upper uterine segment at the junction of the fundus and the body of the uterus in the posterior wall. Right. Sometimes what happens here is the implantation occurs here in the lower uterine segment. Usually the implantation occurs in the upper part of the uterine segment. Sometimes the implantation occurs here in the lower uterine segment, and sometimes the implantation occurs in the abdominal region also. In the abdominal region also, that is called abdominal implantation, and these are the abnormal sites of implantation. Uterine implantation can take place, abdominal implantation can take place, and here the implantation takes place in the lower uterine segment. These are the different sites of implantation, but the normal site of implantation is here, which is at the the junction of the fundus and the body of the uterus. Okay, so that's about the some abnormal sites of implantation, and placenta previa about its 4 degrees, and about the placenta.